Is it on? Yes, it's on. And I just keep an eye on the time. Thank you very much for the introduction. So yes, I think several of us in this room go back to Bangor and North Wales, in particular Gwyn. I can't see Gwyn anymore, who is responsible for me being here tonight, I think. So we'll see. So I'm going to talk about bioelectronics and discuss briefly some of the new developments that have occurred in the field of diagnostics. And there's a tenuous link with smartphones, but it's a great title because everybody thinks, oh, smartphones and diagnostics. So we'll see what the link is as I go through. So the story really is around diagnostic systems and how we can miniaturize diagnostic systems, because probably all of us in this room have been to the GP and they've sent samples away to hospitals, and then you wait and the sample comes back, and usually by then you're better anyway most of the time. And there's been a big drive to try and take all of this analytical capability down into a single platform in order that you can diagnose or monitor health conditions in a number of different ways. And why would we need to do that? Well, you can make quick decisions in GP surgeries, you can perform remote monitoring somewhere in the world, even in space. So NASA have a lot of these devices out in space. If you're out in space for a long time, you need diagnostics, resource limited settings, and potentially home monitoring. This is a project that we had with Philips. I won't explain in details, it never made it to the market for many reasons, but this was for aiding patients monitoring chemo at home. And the kinds of things that we might want to measure are things like full blood count. So full blood count is the commonest test that takes place in the NHS, and that really is about counting the numbers of different kinds of cells that you have in your blood, the numbers of white cells, the number of red cells, and the numbers of platelets. Now you have to send the bottle away, and you get all this data back. Could you do that quickly at home in a GP surgery, for example? We're probably all familiar with the COVID test. That was really the first time we did mass testing of people. These tests actually weren't very good. Mostly they didn't work unless you did them properly, and a lot of people found them very difficult to use. And of course there were tests that were done in central laboratories, but then you had to send the sample away and wait 2 or 3 days for the answer to come. There are new tests on the market now. This is a rapid influenza test, and we can now test for STDs in clinics using these diagnostics. So this field is beginning to develop slowly. Some would say not fast enough. And so we call this a laboratory on chip, or lab on chip for short, and it's a field that's evolved over the last 20, 25 years, really. And this is the sort of text that goes behind that, shrinking and combining analytical techniques onto a single chip and moving stuff around. And in an analytical lab, the stuff that you move is fluid, liquids, could be a sample or salty water, and the stuff that's inside there, the cells and the material that you need to move. So that's moving fluids and particles. And then we can do analysis using optical and electrical methods. And so, this is the schematic that shows that. And often what you find is these devices are pretty cumbersome because there are pipes going into them and wires going into them and so on and so forth. If you can do it, you can make things cheaper, you can make things faster, you can do multiple assays at the same time. Because it's very small, you use a lot less reagents, and they can be very expensive reagents. You can make them throw away, although often that's not such a good idea these days. And you can automate and integrate these devices into other systems. So I thought I'd start with a short engineering lesson in length scale. So most of us have probably got a hair left in our heads, right? So if you take one of these hairs, just give you a scale for size, that's about 0.1 millimeter. So you put 10 hairs together, and that's 1 millimeter. It's a really good measurement if you've lost a ruler. And you can see a hair, right? So 1/10 of a millimeter, we call it 100 micrometers. Okay, if you go down by a factor of 10 only, you have a blood cell, but you cannot see a blood cell. It's too small, and for other optical reasons, it's very difficult to see. So that's a factor of 10. If you go down a factor of 10 again, we're talking about bacteria. So our body has got trillions and trillions of bacteria in our eyes, in our gut, everywhere. You can't see these either. And then down again on the length scale, and we're down to virus particles. COVID was the virus particle, for example, and that's in the sort of 50, 100 nanometer length scale. When we're talking about these lab-on-chip devices and moving liquids around, we're looking at moving fluids on that kind of scale So micro and nano and even picoliters. So that's a microliter is about a cubic millimeter. A nanoliter is 1,000 times smaller in terms of volume, because we've gone down by 10, 10, 10 in each dimension. And the same with picoliters. So picoliters are very, very small, but that's really the volume of a blood cell. So those are the kinds of dimensions we need to make our devices in the sort of 10 micron, 20, 100 micron dimensions. And if you remember this, you'll have learned something tonight. And as I go through my talk, you'll see these dimensions appearing. And so the devices we make are quite large. They're not small like semiconductor chips. And the state-of-the-art semiconductor chips now have dimensions that are smaller than viral particles. And that's something to bear in mind. Whereas we're looking at devices that are fairly large because we need to move large things around. We need to move cells around and liquids around. So they're actually fairly simple to make. So that's a sort of illustration of some length scales. And the moving a liquid on the micro scale is actually more difficult than you might think. If I pour milk into my tea or my coffee, I don't like coffee, then the action of pouring the milk in or moving the spoon around creates what we call turbulence. So we stir the liquid and we can mix it very quickly. On the micro scale, you cannot do that. You can't get a teaspoon in there to stir the liquid. And so if you take two liquids and flow them side by side in one of those devices I showed you, in fact, they will continue to flow side by side for some considerable distance because there's no way that they can mix. And that also poses challenges. And this is a real image here. I think that's about some millimeter, I think. Well, red and green liquid flowing side by side. So they don't mix. So you have to think of ways of mixing fluids on that micro scale. Another interesting thing is we are familiar with inertia. If we dive into a swimming pool, we carry on for some distance before we either drown or we come back to the surface, right? So that's called inertia. If you play snooker, whack a ball, that's inertia. Actually, on the micro scale, that doesn't happen. So for a bacterium or a cell on that scale, it sees the liquid as sticky honey. So as it moves, it has to keep moving. Well, bacteria have these little things called flagella that make them swim. And they use these flagella. And if they stop using those flagella, they will stop moving instantaneously. So there is no inertia on the micro scale. So the physics is very different. And we have to deal with that and think about ways to manage that. So there ended the physics lesson. And now I'm going to go talk about some of the technologies that we've developed. And I showed you this image before. This is one of our very old images of a device that we made. So you can see here there are lots of pipes coming in, providing the liquids, taking the liquids out. This is an analytical device. What we really need to do is to make a hands-free system that allows us to do that. And to do that, we use a technique which is called digital microfluidics, and that will become apparent what I mean by that in a moment. And this is a technique that moves, instead of moving liquid down a pipe, which you're all familiar with, think about garden hose, etc., it moves a droplet of liquid around. And if you can move a droplet of liquid in free space, for example, you can think of that a little bit like a test tube. A droplet on its own, like a marble, contains everything that you need to process. So this is about moving droplets of liquid electronically. So I'm going to introduce you some more physics. Now this is physics behind a technique called electrowetting. So if I take a cabbage leaf, or some plants are very good at this, and drop a droplet of water onto that surface of the leaf. Many plants have hairs on them and the leaf just doesn't wet, right? The leaf balls, the droplet doesn't wet. So it balls into this little ball there. And that stops the leaf getting wet, and that, we call that hydrophobic surfaces, a water repelling surfaces. And you can buy fancy windows now that are hydrophobic, self-cleaning and so forth, right? That's a hydrophobic surface. Now, it turns out that if I take a thin piece of metal, and coat that surface with a very thin layer of Teflon, like a frying pan. Okay, so this is a metal electrode, and put a drop of water on there. That will also be hydrophobic. You know that. If you buy a brand new frying pan, it's hydrophobic. It's got Teflon on it. Well, what now if I apply a voltage to that? So in this cartoon here, what I'm doing is I'm applying a voltage. This is a battery. Between this bottom surface, which is metal. And to complete the circuit, I put a small piece of electrode or wire into my droplet of water, and I apply a voltage. It turns out that I can use this voltage to change the way that that droplet behaves, and this is what this video here shows. So I'm turning the voltage on and off, and I change the shape of that droplet. Okay, so that's called electro-wetting, changing the wetting properties of a drop of liquid. And you can do that, and actually many years ago I was working with Philips, and there was this idea that you could create an infinitely variable lens using this kind of idea, right? Didn't really take off, but there are companies, actually Pilkington now make a lens based on this idea, because just by controlling the voltage you can change the shape of that lens. Exactly. So this concept is called using electric fields or electricity to change the wetting behavior of a droplet, electrowetting. Now that's a droplet. Remember I was talking about digital microfluidics, this droplet that we want to move around. How can we actually do something with this droplet rather than just keep it there and look at this wonderful video? So how do we control, this is where the link goes with the mobile phones, how do we control hundreds of single droplets? So we need hundreds of single electrodes, right? If I go back one slide, you can see this is an electrode. If I have hundreds of electrodes, I begin to be able to process hundreds of droplets. And the where you might find hundreds of electrodes actually are in the screens of mobile phones. So you probably can't see this from the back. And I would normally ask this question, how many people here have been curious enough to put their mobile phone under a magnifying glass? Probably nobody at all. Ah, there you go. One person. Excellent. Two people. When you ask school kids, they all put their hand up. We're not curious enough. So what you see, if you put your mobile phone, not the really modern ones, under a microscope or a magnifying glass, are lots and lots of little dots. And these little dots are called pixels. And each of these dots, in fact, is a single electrode. And we apply a voltage to these different dots, these different pixels, to create the image. Right? So each pixel will have a different voltage on it to create the image. And what the voltage does is controls a liquid crystal. I'm not going to tell you about that. But actually, this screen provides us with a platform for creating voltages to hundreds, thousands, in fact millions of individual electrodes. And so I was lucky for 5 years I worked with Sharp. They've closed now. They had a laboratory in Oxford where we were taking these screens that exist in mobile phones and using them to move droplets around. And so this is the repurposed screen. You can't just take a mobile phone and take out the screen, but you use the same architecture, and we made these in Japan, re-engineered them so we could use the electronics to move these droplets of liquid around. And you create the device which has got 100,000 individual pixels or electrodes, and then you can move hundreds of droplets around. And this is a cartoon showing one of our early devices, and you can see these little dots are electrodes, and on top of each of these dots there sits a ball, right, a droplet. Big droplets outside, these are our reservoirs, and then we control the electrodes and move the drops around. So I'm going to show you in the next video, which is an animation, a cross-section of that. So you're going to be looking in this direction. And then if I switch that on, you can see if I turn this electrode on here, I move the droplet to there. And if I turn that electrode on there, I move the droplet to there. And the gap between the bottom and the top here, right, so there are two sheets of glass, is 100 microns. That's a human hair, right? So the size of our droplets are about 100 microns, about the size of your hair, okay? So in a conventional mobile phone screen, look at my pointer, we would be looking down that way through the top glass, and the electronics is behind in the back, okay? So there's our electrodes, there's our Teflon, and there's our droplet, and we actually have oil in there. And this is a video that was made by my colleague Ben in Sharp. Now, I thought I'd show some of this video. It's quite long, but this is actually our first devices. And what you're looking at here is one of these devices that were made by Sharp. And we've got a microscope here, and we're just looking through the microscope down onto the top of that device. And each of these little patterns is an electrode. Doesn't look like an electrode, but it is. And there's a scale bar there, 1 millimeter. And because you've got those two sheets of glass, these droplets have now become more like flat pancakes, okay? Because you squash the top and the bottom. I'll play some of the video because what you can see is just by changing the voltages, you can move drops around, right? So we can make a bone-shaped structure. We included into the device electronics so we could actually, instead of looking through the microscope, read out electronically where the droplets were. So this is an electronic image of the droplets, still an electronic image of the droplet. You can do things with a mouse, you can click and you can say, I want this droplet to move over there. That droplet I want to combine with that droplet. There's a whole bunch of stuff you can do, and this is all now, as I said, digital fluidics done on this small device. This is dispensing droplets. This is the sensor. This is not optical. This is optical. This is electronic output from the device. And we can split droplets. So this is showing the optical image of we split the droplets, and so you can play all sorts of games with this, which is fun. But the question is, what can you do with it? Any company that's going to make these devices is going to think, actually, in the end, we need to sell these and make a profit. So I'm going to take a diversion and talk a little bit about DNA sequencing. And you probably all heard about sequencing during the COVID pandemic, right? We were all sequencing our COVID viruses. So what is sequencing? I'll tell you in the next slide. But sequencing really took off, and you all probably were aware of the Human Genome Project in 2003. This was a remarkable feat, really. It took 13 years. Cost 2.7 billion to sequence the first human genome. It wasn't a genome from a single individual. It was, in fact, they only finished this about 3 years ago. It was pooled DNA from lots and lots of people. But in 2008, 5 years later, a single human genome was sequenced in 5 months, and it cost $1.5 million. So 2008, that was not that long ago. It now costs, I'll put this in dollars, around $150 to sequence a genome, and it's routinely used in clinical diagnostics, particularly in cancer, for example. You're sequencing your cells to understand what they're doing. And that's really remarkable. 2003 to 2024, we've gone from these kinds of figures down to something that costs £100 to sequence. So it's quite complex, but I thought I would tell you a little bit about sequencing. But before that, you all remember this chap? I'm not going to make any comment but about him, but in 2018, I think it was, he said all children should receive whole genome sequencing at birth. Probably not a good idea because there are all sorts of ethical issues there. Who owns the data? Where does the data get used and so forth? That didn't actually work. But now that's been reborn, and you can go online and see this Newborn Genomes program. It came out this year. You opt into this. It's about sequencing the genomes of 100,000 babies, looking for genes linked to more than 200 rare conditions. So actually, it's very, very powerful what we can do by looking at the human genome. So how does sequencing work? Well, DNA is the book of life. Really, that is the instruction that our body and all the cells in our body uses to make the proteins. And there are 4 letters in that DNA, GATC. Okay, we'll probably all know that, and if you don't, well, that's something for you to remember tonight, GATC, right? So those are the letters in the genome. And the genome is the double-stranded DNA, which gets wrapped up into chromosomes inside the cell. In us, there are 3.2 billion letters, and sequencing is about reading those letters, reading that sequence of letters, because these are all, apparently they're random, they're not random, but you know, you read a G and then a G and then an A and then a T and so forth, and that's the sequence. And there are a number of ways of doing that, but one of the revolutions came from this company in Oxford called Oxford Nanopore, and it's called nanopore sequencing. And this is about using electricity. So we're back to bioelectronics to read these letters. And the way that this is done is they have an insulating structure, which is this block here. And in that insulator, there's a very small hole, and that hole is made by a protein, shown here in brown. And the DNA is, we know it's a double strand, it's split into a single strand, and as it moves through this hole, we measure a current. And amazingly, that current will change depending on you have an A, a C, a T, or a G. all right? And it goes through. Imagine that you could think of this like pearls on a string or something. You can read an A, T, C, G just by measuring the height of that current. And this is what Oxford Nanopore in Oxford invented and developed, and was used very widely, still used for sequencing. And it's so small that you can hold it in the palm of your hand. So this is the device, not this, but this is the sort of size of a device that Oxford Nanopore now manufacture. It's USB. You plug it into your computer, you do the chemistry, and you can sequence DNA. And they made devices that do this, but this particular device, called the TrackScient, combines that digital microfluidics I was telling you about for doing sample processing, because you can't just take blood and put it into your machine. You have to process it, you have to extract the DNA, throw away a lot of rubbish and so forth. And this digital microfluidics does a lot of that. So the video I'm going to show you now tells you that story and how it works. And the sound is terrible, so just look at the subtitles. So this is about preparing the DNA. So here you see the DNA. There are some things on the end. This is that hole I explained to you. This is how the DNA goes through the hole. And it's ratcheted through. And so you get real-time analysis of the electrical current, right, back to bioelectronics. And the machine that does the sample processing is the machine that we developed with Sharp. It's called Voltrax, and Sharp marketed and sold this. And if you look in this video, you can see these small droplets being moved around on this device, and this does the sample preparation. They call it sample prep, sample cleanup, ready for the DNA sequencing to take place. And these are tiny little machines, and basically this is about the size of your mobile phone screen. So it shows you how you do the chemistry and so forth. How are we doing for time? And it's a remarkable piece of technology, the nanopore sequencing. People have taken it into space, up Everest, underwater, and you can sequence DNA anywhere and everywhere. So you can see the droplets moving around and complete electronic control. So I'll just move on a little bit. Whoops. So if you can sequence an individual's genome, you can then begin to think about things that are called, or techniques called personalized medicine. So we kind of know now that we hand out pills and expect everybody who takes that particular pill for a particular ailment to have the same response, right? But that's really not the case. And tailored or personalized treatment is certainly already there for cancers, but it's becoming much more interesting for a number of other diseases. And so what you can do is, you diagnose an individual not just based on a physiological exam, but on their blood, for example, on their, I can't really read this, so you probably can't read it, but on their genomics here, and proteomics and molecular properties. And this is what we call omics, so this sequencing that I've just been speaking about. And there's a spelling mistake there. But it really requires development of many then different proteins, different products, so that you would have a very different medicine from me and other people. And that, we already see the beginning of that in immunotherapy, for example. Cancer vaccines, they are developed for a subset of the population and not for everybody. Patient-specific antibodies, growth factors, hormones, factor VIII, for example, was a long time ago that was developed, enzymes. So we need to find ways of making proteins that are specific to an individual's physiology and makeup and disease. This is called targeted treatments and personalized medicine. So proteins, you've all had the flu jab, I guess. Many of you had the flu jab. So the flu jab is made by injecting antibodies into a chicken egg. That's how it's usually made. But now that jab is made using cells. So we stopped using chicken eggs, we now use cells. And these cells are grown in things called bioreactors. So here we have a bioreactor. It's a flask, and in that flask you have lots of cells, like maybe cells from muscle cells, for example. They're called fibroblasts. You don't use muscle cells, but for example, cells like that. And these cells make proteins. You can program them by using the right DNA to make proteins. This is a very expensive process. It's fine if you want to make a flu jab because you sell hundreds of millions of the same jab. But if you're making a tailored protein for me and a different one for you, then this is really not the way to do it. It's too expensive. And so there is a technology that's been developed, and it's remarkable when you think about it, which is called cell-free protein production. So a cell makes a protein because it reads the DNA, and it says, this gene, I need to make this protein, and then I use that protein for something, right? Maybe for my muscles or for digesting food. It turns out, actually, you don't need the cells. If you mash up the cells and extract the machinery, and you add DNA and a little bit of other things, you can make the proteins without the cells. And this is called cell-free protein production. Cell-free protein production. And that's remarkable if you think about it. You don't need the machinery. The cells, all you need is the bits and pieces inside the cell. You take it out, throw away the cell is dead, but the machinery still works. And so I worked with a company in Cambridge that used the same platform I've just shown you, but we scaled it up quite a lot. And this is the device that we use now, so it's much, much bigger. This is also like a mobile phone screen. It's a cartridge. We put it into this machine, and this cartridge takes a particular DNA sequence, and I've told you about DNA. You mix it with this cell machinery. We don't need the cells, just the machinery. Incubate it in the droplet, those droplets I've spoken about, and you wait a little bit, and then you purify proteins at the end of the day. So you can make proteins in this machine, and that takes 24 hours to make nearly 200 different proteins. And we do it using the same idea, but on a bigger scale, if this video works here. So this video is showing these droplets moving around on this platform. You can kind of see the scale of it, because I just held it up here. So you pipette the droplets in the side, and then all of the mixing, all the processing, all the cooking is done on this piece of glass using that digital microfluidic technology. And you can see these droplets moving around. And we're basically writing images to this piece of glass, this piece of electronics, continuously, just like an image on a TV screen, to move the droplets to where we want them and to do this processing, mixing.
Sorry, Howell, is that an animation or is that real?
No, it's real.
That's real.
Yeah, it's real. It's a bit speeded up, but it's real. And to get this to work, in fact, Philips, unfortunately, the lab in Oxford closed. So this is much bigger than the one I showed you originally. We worked with a company in Boston that I doubt any of you have heard of, E-Ink. How many of you got an Amazon Kindle? Yeah. Right, lots of people, right? So E-Ink make the Amazon Kindle. And if you think about it, the Amazon Kindle is actually very much like a mobile phone. What it does is creates an image. And then we're using the Amazon Kindle hardware to create these images, and these images are moving these droplets around. And so it's a repurposed Amazon Kindle. Just keep an eye on the time here. So that's a technique that can manufacture tailored proteins on this platform. These proteins are not good enough to inject into somebody's arms. These proteins are designed for pharma companies to figure out different shapes, different sizes, how they work, and then they scale them up. So it's a rapid way of doing pre-production experiments to determine which proteins are best to use. And it uses DNA and this cell-free, it's called expression, cell-free protein production method. So I have a few slides. I'm just going to have a little swig of my refreshment. To sort of finish the next 15 minutes to talk about something which you probably all are familiar with and hope you never have to deal with, which is antimicrobial resistance. Some say this is the next global pandemic. So antimicrobial resistance has been around a long time, ever since penicillin was discovered. Within a few years, we already found bacteria were resistant to penicillin. It's grown into a big problem now. This is a report that was published by the government this year, Confronting Antimicrobial Resistance 24-29. It's full of useful guidance and tips and so forth, but you know, this is a big problem. And you look at these headline figures, it causes globally 1 million deaths per year, and it's a contributing factor to a further 5 million deaths annually, which is more than the combined burden of influenza, TB, and HIV. By 2050, these statistics are probably 10 years old now, and we're on track to reach this. It could be the primary cause of death with 10 million lives lost per year, more than cancer. Right? And if you go into hospital, you have probably between a 7 and 10% risk of catching an infection in hospital. And of those people who catch infections, a number of those are resistant to antibiotics, and then that leads to things like sepsis and high mortality, morbidity. So there's a quiz to sort of get you going a bit. So this is a map of Northern Europe showing resistance rates. You don't need to see the color code, but red is bad, green is good, for 4 different antibiotics. 1, 2, 3, 4. This one's penicillin. This one is a fluoroquinolone, doesn't matter. This is a third generation cephalosporin. This is a carbapenem. This is one of the antibiotics that is called antibiotics of last resort, carbapenem, meropenem. And you can see that penicillin resistance is probably nearly 100% now, right? So we don't use penicillin anymore. And Scandinavians, pretty good. Most of the countries over here that are not on this map, places like Bulgaria, Romania, are absolutely awful. But you can see how things are depending on the kind of antibiotics. So fluoroquinolones, we are generally better than the Spanish, the Italians, and so forth. Part of that is to do with the way that antibiotics are prescribed in these countries. So until recently, you could go to the pharmacist and buy antibiotics over the counter in Italy and in Spain. Italy is particularly bad. These are third-gen cephalosporins. And these are carbapenems. So the question is, which one would we use if you were a GP? Some of you may be GPs. Are there any GPs in the room? Luckily there aren't. So oh, there is one. So, okay. Which one would you use? So there's no right answer to this. You're all very shy, worse than children. So you'd all say, we'll use this one, carbapenems. Actually, you probably wouldn't, because it's very expensive and it's got really bad side effects. That's used in hospital ICUs. So probably a fluoroquinolone, a cephalosporin, but actually it's a very difficult decision to make. You don't know, as an individual, which bacteria that you've got, which antibiotic will treat the bacterial infection that you may have. Right, and this is real data. You can go on, if you're really bored one night and there's nothing on TV, you can go on this website and look at all this data and download it. So usually, if you give somebody penicillin and they get better, they probably haven't got a bacterial infection in the first place, because we're so resistant now. This one, the carbapenems, meropenems, we don't really dish that out because it's £2,000 or £3,000 a pop, and you don't want to develop resistance to carbapenems because this is our last antibiotic that's in the cupboard, right? But look at this, third general cephalosporins, 2004, 2019, right? So Italy in particular. It's a real problem. And so there are a number of issues that one has to deal with to try and resolve this. First of all, stop giving antibiotics out. GPs give lots of antibiotics out for many reasons. It got worse during COVID because so many people were on incubators and having lots of antibiotics that resistance rates went up. And we all remember David Cameron. When was Cameron? 10 years ago. He asked Jim O'Neill, who was an economist, Lord O'Neill, to look at the issues around antimicrobial resistance from the perspective of an economic argument rather than just a medical argument. And he wrote an excellent report tackling drug resistance. And he made a whole list of recommendations. You know, some very simple ones, stop using antibiotics in agriculture and so forth, which we're pretty good at in this country. And number 5, promote new rapid diagnosis. Support the use of rapid point-of-care diagnosis to facilitate the mandatory use of tests to support clinical decision-making by 2020. So where are we now? 2025 in a few weeks? That hasn't happened. One of the reasons it hasn't happened is it's very much easier to give a pill out because it costs a few pounds than it is to do this diagnosis. The other is there aren't that many diagnostic tests around. And so we've been working on this problem for a number of years. But I thought I'd sort of say to you a little bit about how testing is now done. This is a sample of an infected urine. Somebody has a UTI. And yeah, it does look nice, doesn't it? Yeah. Probably women will have 2 or 3 UTI episodes in their life, men maybe 1 now. And it's becoming a real problem, UTIs, because more and more UTIs are becoming drug resistant. And many elderly people are on prophylactic antibiotics. But okay, so you go to the GP, you give a sample. After that goes to the lab. So what do they do in the lab? They take this sample and they scrape it onto a plastic plate that's got some growth agar on it, right? It's like a jelly on which the bugs like to grow. On day 2, you can see the bugs. We call these colonies. There are lots of bugs growing. You haven't done any diagnostics yet. So what we do then is we place little discs. Can you see these dots? These are discs impregnated with antibiotics onto a new petri dish, and you can see the bugs have grown everywhere. But depending on the disc and the antibiotic, they'll stop growing closer and closer to the disc, right? So around this disc here, they've stopped growing. That's the zone, it's called zone of clearance. Can you see these zones, right? And so what they do in hospitals, they take a ruler and they measure this zone of clearance, and they look up in a chart and say, well, that's the concentration, and that bug is resistant or not to this antibiotic. So if it's resistant, there will be no zone in, it will grow right up to the disc. This hasn't changed in 100 years. We're still doing this. And then you get stacks and stacks and stacks of these dishes in a lab. And then maybe on day 4 or 5 or 6, the results go back to the GP, and you'll have, well, you probably won't have a consultation, because either it will have been resolved or it'll have got a lot worse. So this is called a phenotypic test, because you're looking at the phenotypic response. This has got nothing to do with genes. This is the phenotypic response of the antibiotic. Has it killed the bug, or is the bug able to survive? And the lab workflow is about 3 days. And on the one hand, the government is saying stop using antibiotics and we need to have these tests. And then recently, the government introduced a new way of accessing antibiotics. So if you have a UTI, you can now go to pharmacy, they give you 3 days worth of trimethoprim and hope for the best. So these two things are completely opposite ends of a spectrum. And the GP will say, you've got a UTI, you probably need nitrofurantoin on trimethoprim. It doesn't work, you have to come back, you do more tests. Unfortunately, then things can go wrong, patients can die, resistance increases. So we need better ways of dealing with this problem, right? And this is just the UTI story. If you have sepsis, it's even worse. So rapid tests need to be, if you're in hospitals, because actually GPs are too busy to do this, it needs to be done within, people always think, oh, it needs to be done in 10 minutes. You don't, but it needs to be done at least within a clinical shift. Right, so rapid, this means not like a glucose prick, but something that takes maybe 4 hours or something sensible, not 4 days. And so we have developed a new test in Southampton. So it's called a rapid antimicrobial susceptibility test. Is the bug susceptible, or is it resistant to the antibiotic? It's important to say this test does not tell you whether you have an infection that is viral or bacterial. It tells you whether or not, if you have a bacterial infection, whether the antibiotic is going to work. And if this test is done properly, instead of a guess, a finger in the air, it will tell the physician the correct antibiotic to prescribe right at the beginning. And that reduces the test time from a few days to 3 to 4 hours. And so how does that work? Well, we have these little devices, these little chips that we make in university, and we pump liquid and cells along a channel. So the chip has got a channel, And remember the dimensions I spoke to you about at the beginning? So these are bacteria, they're about a micron in size. And the dimensions along this axis is about 20 microns. So it's 2 cells. So these are a bit smaller than the human hair. And inside this channel we have some microelectrodes. That's what these black things look like here. And we apply a voltage, and these lines are lines of electrical current. And the bacteria are pumped between these electrodes, and they disturb the current, and we measure an electrical signal. So we measure the electrical properties of single bacteria quite quickly, high speed, in this chip. And we expose bacteria to antibiotics for 2 hours. And then we have an unexposed population. So we exposed, unexposed. And we measure the electrical properties of the bacteria one by one very quickly, takes a few minutes, and compare exposed and unexposed. And if the antibiotic affects the bacteria, we get a different electrical signature. So watch this idealized, I don't need to do that, watch this idealized bacteria. I'm going through here, and now I'm going to introduce the antibiotic, and you'll see that the shape of this bug changes slowly. This is speeded up. There you can see, right? This little bulge comes out. The bulge gets bigger and bigger. And if the antibiotic is working, eventually it kills the bug. So it forms this thing called a spheroblast. And then we get these fragments. So that antibiotic in that cartoon has worked. And that actually only takes about half an hour to measure. So it's a very quick test. And the kind of data that you get, we call them scatter plots. Each dot in this fuzzy picture here is a single signature, or a single signal from an individual bacterium. So this is our control that has not been exposed to the antibiotic. And here, after exposure, susceptible, this one, the dots move. There they were originally, and now they move somewhere else. And that's all we need to do, is look at where these dots have moved on this graph. And in fact, we can measure 10,000 bugs in about 30 seconds. So it's a very fast test, 2 hours incubation, and a 30-second measurement time. So the next slide shows this happening in real time. So these are the dots appearing on the screen. This is our control, no antibiotic. This is trimethoprim for a UTI. And this is ciprofloxacin. And you don't need to be a genius to work out that actually these bugs are resistant to trimethoprim because they're exactly the same as the no antibiotic. And these bugs, they've all disappeared because they're susceptible to ciprofloxacin, right? So ciprofloxacin, lysis is a fancy word for explodes the cells. So these cells are susceptible, so all we get here is debris. The resistant cells are unchanged. So trimethoprim is a common antibiotic that's given for a UTI, and in this particular case, these were data on UTIs. So this would have been resistant to trimethoprim. So guess which antibiotic worked? This is data we collected with our friends in Porton Down, UK Health Security Agency. This is our control sample. This is trimethoprim, colmoxiclav, amoxicillin, gentamicin, ceftazidime, cephalexin, ciprofloxacin, and nitrofurantoin. That one is clearly susceptible. It looks completely different from the rest, right? So this is a fast test and says, actually, if this individual had been given the common one, trimethoprim, she would have come back and said, it doesn't work, and then we'd have given them nitrofurantoin. So this is data that we collected with colleagues in Southampton. And this patient had a bloodstream infection, which is very serious because it's a very high percentage of people with bloodstream infections unfortunately die. And you can see here, same kind of data, resistant. Meropenem, do you remember that antibiotic I mentioned 10 slides ago, the green one? So that's susceptible to meropenem. Resistant. Right, so that was done in 3 hours. And I keep walking over here. That one is susceptible, so that's the one you should use. The lab result came 2 days later, and it said it was susceptible to meropenem. So it shows that this technology is very simple, but actually it works. Our result was done in 3 hours. And we formed a company. So 3 years ago, this was the kind of box we had on our bench at the university. see the electronics and the plastic chips here. And now we have a company, and this is where we are. This is the device with. It looks big. It is big because we've got robotic systems in there. So the chips are quite small, but you've got to do all the fluid handling and so forth. And we've done tests in Dorset, QA, Southampton. We're about to launch in the UK market at the end of next year. And also in the US, we have to do this FDA approval, and that's in progress as well. So I just thought I'd finish. I think, yeah, bang on time. Why next for microfluidics? So I've talked about diagnostics. Well, the quest really for microfluidics is moving into different domains. So, and this is about something we call organs on a chip. And many of you think, oh, organs on a chip, that's about reproducing or creating organs for transplanting. It's not. It's completely different. This is about recapitulating, reconstructing the form and function of human organs outside the body, so that you can take things, I can't read this, so you can't read this, like mouth, bone, brain, liver, spleen, etc., and grow these organs from cells outside the body in small devices, microfluidic chips. And if you can do that, that actually makes science so much easier, because you haven't got to do experiments on animals. It's very difficult to do experiments on humans. You can very easily access the material, and the benefits are amazing, because you can begin to understand disease in many different ways, develop all kinds of different drugs. I worked with colleagues looking at asthma analogy, and if you grow, you can grow sort of, shall we call it inverted commas, lungs on chips. The behavior of somebody, of these organs, if you take samples from somebody who has asthma or somebody who's normal, is very different. So you can begin to think about studying, understanding, and developing personalized medicines for people who have different kinds of ailments. And we call these in vitro models. And so we're going from human disease in humans to models on the bench. I thought I'd leave you with this wonderful cartoon, which is on this website, which shows where people are going. And this is all about trying to recapitulate an entire human on a device, on a chip, where you've got the heart connected to the lung and the kidney and the spleen and the gut and so forth. And people are moving into this field now. And you can study things like immune response, transport, metabolism, and so on and so forth. And I just want to really finish by acknowledging a lot of my colleagues. I've worked with lots of companies and very clever people, particularly Ben in Sharp, Dan Spencer at the university. My company, Toby King, is our CEO, colleagues in Nuclear, Yink, and other companies and funding agencies. And so I'm very happy to take any questions.